Hello, class. My name's Lavi. I'll be doing my research project on the Zika virus. So I'll be discussing the article that I found and what was discussed within the article. So to start off, the Zika virus is spreading throughout more than 20 countries in the Americas. Mothers grow worried for the future of their children and the future children. So the type of research conducted in this study is qualitative and descriptive. Presented in this article was the vertical transmission between the mother and the fetus, and how the Zika virus transferred to the fetus. Um, so during the first during the end of the first trimester of the subject um, is where they think the Zika virus was transmitted to the fetus. She was in Brazil at the time. Um, she's 25 years old female, and she presented with a fever that was followed by severe musculoskeletal and retroocular pain and itching. So she also had a generalized rash along with that, and this was during the 13th week of gestation. Um, so sample attributes of the study include the autopsy and examination of the central nervous system. The research findings showed that there was severe fetal brain injury associated with the Zika virus through vertical transmission. So the scientists performed the study to find a link between the Zika virus and microcephaly, which is the problem now. Um, so possible connections between the pregnant woman with babies having microcephaly and the Zika virus are obviously connected. We've seen it in the news. So methods that were used in this specific research included um, an autopsy of the central nervous system, as I mentioned earlier, um, electron microscopy, indirect immunofluorescence, and microbiologic investigations. So with the autopsy and central nervous system, an autopsy of the fetus and the placenta was performed 3 days after the termination of the pregnancy. Extensive sampling of all of the organs, placenta, the umbilical cord were done. All of the samples were fixed and buffered formalin and embedded with paraffin. The tissues were collected for microbiologic investigations. Um, so the brain and the spinal cord were also fixed, but with a higher percentage buffered formalin for 3 weeks. So I'm thinking it was done with a higher percentage because they're bigger organs. So following this was a neuropathological examination that was done and performed with extensive sampling of the brain and spinal cord. So sections of all of the tissue samples were stained, immunostaining for Gilead fibrillin acid, protein, neurofilament, human leukocyte antigen, DR. This was used to detect a specific protein in the sample, to see if it was the Zika virus that caused the microcephaly. So the CD3 highlights the T cells in the samples, and the CD20 highlights the B cells in the samples. So these were performed on representative central nervous system samples. The electron microscopy, this is another method that was used throughout the research. Tissues were collected from formalin-fixed brain, and it also went under additional fixation. Semi-thin sections were made stained and analyzed by means of light microscopy. Ultra thin sections were stained as well and analyzed. Negative staining was also performed. Um, Imaging of the ultra thin sections and brain were performed with the use of a plus transmission electron microscope. So, with the indirect immunofluorescence, the paraffin embedded sections of the fetal brain tissue and brain tissue of the autopsied man, which they used for negative control, were incubated with serum obtained from the mother of the fetus. Okay, so in addition, the fetal brain tissue was incubated as well with the serum obtained with a healthy blood donor, as well as with labeled anti-human antibodies only. So the microbiologic investigation, the RNA was extracted from the placenta, the lungs, the heart, the spleen, the thymus, liver, and kidneys, and the cerebral cortex with the RNA purification kit. The next generation sequencing was performed in samples of the fetal brain tissue in the use of an ion torrent and Genius software. So for the paleogenic analysis, complete genome of the Zika virus sequences were used. Multiple sequence alignments were performed. A detailed description of the molecular methods were provided in a supplementary appendix, which is available with full text of the article at negm.org. All right, so for the results of this article or research study, so let's see here. So the findings in the autopsy and neurological examination, the fetal body weight was in the 5th percentile. The length was 42 centimeters, which is in the 10th percentile, and the head circumference, which was 26 centimeters, was in the 1st percentile. So the only external anomaly that was noted was obviously microcephaly. The placenta weight, which was 200 grams, resulting in the placental fetal weight ratio, which is below the 3rd percentile. So the microscopic examination of the central nervous system revealed microcephaly with a whole brain weight of 84 grams below average. So that's really small. There were also numerous variable sized calcifications in the cortex and subcortical white matter in the frontal, parietal, and occipital lobes. The subcortical nuclei were quite well developed. The microscopic examination revealed appropriate cytoarchitecture of the brain, of the fetal brain. Activated microglial cells and some macrophages expressing human leukocyte antigens were present throughout the most of the cerebral gray and white matter. So this is a sign that the body knew that there was an infection there. So they also had scattered mild perivascular infiltrates composed of T cells and B cells that were also present in the subcortical white matter. This is also a sign of infection or something that has entered the body that's not supposed to be there, a foreign object. Okay, so the cerebellum, brainstem, and spinal cord showed neither inflammation nor dystrophic calcifications. The brainstem and the spinal cord showed degeneration of the long descending tracts, especially the lateral corticospinal tract, whereas the ascending dorsal columns were all preserved. So indirect immunofluorescence revealed granular intracytoplasmic reactions in destroyed neuronal structures, which pointed to a possible location of the virus in neurons. There were no relevant pathological changes in other fetal organs or in the umbilical cord fetal membranes. So results within the with the electron microscopy were clusters of dense virus-like particles of approximately 50, I'm sorry, excuse me, 50 nanometers in size were found in damaged cytoplasmic vesicles. So the groups of the envelope structures with a bright interior were also found. At the periphery of each group's, the remains of the membranes could not be seen. And with the microbiologic investigation, there were positive results of the Zika virus that were obtained in the samples only in the fetal brain sample, where viral RNA copies per milligram of tissue were found. So all autopsy samples were tested, excuse me, and they were found to be negative for other flaviruses such as the yellow fever virus, West Nile, the varicella, the zoster virus, herpes simplex virus. A complex Zika virus genome was recovered from the brain tissue. The paleogenetic analysis showed the highest identity, which was 99.7%, with the Zika virus strain isolated from a patient from French Polynesia in 2013, and the Zika virus detected in San Pablo, Brazil in 2015, followed by a strain isolated in Cambodia in 2010. So with the strain from the outbreak in Micronesia in 2007. So in the Zika virus polyprotein, there are 23 polymorphisms that were detected in comparison with the strain from Micronesia, and 5 polymorphisms in comparison. So my discussion, I chose this particular article from the rest because it discussed the links found between the Zika virus and microcephaly and the fetuses, and actually had a specific case reported associated with it. I found this article to be very interesting because the detailed case study on the subject, this article was very informative and clearly written. I thought this article was very wordy in my opinion, and I would recommend this article to someone with a medical background, specifically with microbiology, because of all of the wordy terminology, and also have a familiar with medical research, because again the terminology used in the article was very very difficult, and I had to look up some of the stuff myself because I forgot some of the terms. The author's strengths of this article is that the link between the Zika virus and microcephaly in the fetus were found. The complete genome sequence was recovered in the study and consistent with the observation that that is present in the Brazil. That is a present strain in Brazil from the Asian lineage. Excuse me. Limitations the author has is that there's only one subject that was used within the research, when it would have probably been a little better if they had multiple subjects that they were able to use. But again, I do realize that it's sort of a new virus that is now up and coming, and probably hard to find subjects that are willing to donate the fetus that was terminated or um, and so on. So thank you again. My name is Lavi, and I hope you enjoyed my presentation.